Hello children, today we'll start with the chapter The Fundamental Unit of Life. So let's learn more about the basic things about cell. All living things, as we all know, are made up of cells. Cells in Latin means a little room. Cell is the smallest structural and functional unit of life. Since the body is made up of cell, it is called the structural unit of life, and since all the functions of the body, whether it is physiological, biochemical, genetic and other functions are carried out by cells, the cells are also known as functional unit of body. There are two types of cells, namely prokaryotic cell and eukaryotic cell. Prokaryotic cell means the cells which have primitive nucleus, that is, the nuclear membrane is not well defined, whereas eukaryotic cells have very well defined nucleus. So here you are seeing the picture of Robert Hooke, the scientist who discovered cell. He saw cells something like this. And these small little structures that he saw, he called it as room. So how did he observe this? This is the microscope that Robert Hooke used to observe the Koch cells. So if you see the different parts, here for light he used flame, and to focus the light he used a water lens, and then his specimen was kept over here and he used to see from this place the magnified specimen. So these are the enlarged structure that he had seen, that is the cells of the Koch, which he called as little rooms, now that we call them as cells. So this is the compound microscope which we generally use now in our laboratories, though we have more higher versions which you will probably use in your higher classes. So a compound microscope has an eyepiece through which you look into. It has a revolving nose piece to adjust the objective. The objectives will have different magnifications, so you will keep the objective near the specimen according to the magnification that you need. This is the stage where you keep your specimen or the slide. And here you have the mirror onto which the light passes. It passes through this condenser and focuses on the slide so that you can see the image through this place. And then you have here an inclination joint which will help to move this arm further or backward, forward or backward. This is a coarse adjustment which will help you to move this whole structure up and down. And this is a fine adjustment where you can make fine, very fine movements so that you can adjust whatever you're seeing according to your eyesight. So if you see the onion peel cells, this is how they will look under microscope. And this is a magnified version. And this is the microscope that you would be using in your laboratory. Let's know more about few discoveries. Robert Hooke discovered cell in 1665. Anton von Leeuwenhoek observed free living cells in pond water in 1671. Robert Brown discovered nucleus in 1831. See here 1671 and 1831. After a very long duration, nucleus was discovered. Purkinje coined the term protoplasm in 1839. Sheldon and Swan proposed cell theory. It stated that all plants and animals are made up of cells. Virchow further suggested that all cells arise from pre-existing cells. So if a cell has to be formed, it has to be formed from a pre-existing cells. Organisms are of two types. One, unicellular organism, and other, multicellular organism. As the name suggests, here single cell constitutes the whole organism, and here many cells group together in a single body, and they assume different functions in it to form various body parts in multicellular animals. Basically, unicellular organisms constitute organisms with single cell, and multicellular organisms have many cells. Here you have example paramecium, amoeba, chlamydomonas, and in multicellular you have examples of fungi, plants, and animals. These are the different types of cells that are found in the human body. Here we can see a smooth muscle cell, blood cell, nerve cell, which is supposed to be the longest cell in the body. This is a fat cell, bone cell, and ovum or the egg. Now, in the previous slide we have seen all the cells were not of the same shape. Different cells had different shapes. So the shapes of cells are related to specific function they perform. Now the nerve cell was long. So why is it long? It is in the brain, right? Nerve cells originate from the brain and then they have to send signals to different parts of the body. Therefore, they are long. In human body, each organ performs a specific function. Hence, the cells of different organs have different shapes and sizes. Different parts of human body perform different function. The heart pumps blood, stomach digests food, and so on. That is, there is division of labor in multicellular organisms. So different organs have taken up different functions. Therefore, we see that multicellular organisms have division of labor. Similarly, a single cell also shows division of labor. Each cell has cell organelles. Cell organelles are the components of the cell, and each organelle performs a special function. We'll study about these special functions later on. So, all cells, whichever organism or whatever function they are performing, they all have the same organelles. The components of the cell remain the same irrespective of the function or in whichever organism they are present. Structural organization of a cell. There are three common features in cell. 1. Plasma membrane. 2. Nucleus. 3. Cytoplasm. And in plant and fungal cells, they have cell wall in addition to the above components. Let us now understand the three structures with help of these diagrams. Here we have the plant cell and here we have the animal cell. In case of plant cell, you observe a cell wall, but in case of animal cell, we do not have a cell wall. Within the cell wall, we have the cell membrane, and enclosing within the cell membrane is the cytoplasm. Enclosed within the cell membrane is your cytoplasm, and this nucleus lies within the cytoplasm, In the nucleus, you have a dark structure which is nucleolus. We will study further about it, right? So what are the basic three structures? You have the cell membrane, you have the nucleus, and the cytoplasm, which is there in any kind of cell. But the cell wall, this cell wall you see only in case of plant cells. Plasma membrane. It is the outer covering of the cell. It separates the contents of the cell from external environment. That is, the cytoplasm is there within the cell and it does not allow the external environment to interact with the cytoplasm because it covers the whole cytoplasm. It encloses the cytoplasm. It is made up of proteins and lipids and is also flexible. It permits movement of certain molecules and it prevents others. Therefore, plasma membrane is called as selectively permeable membrane or semi-permeable membrane. It helps in diffusion and osmosis. We'll learn more about diffusion in the next slide. Diffusion. Movement of substances from high concentration to low concentration is called as diffusion. So if you take plants during photosynthesis, the level of oxygen in the leaves is high. The concentration of oxygen in the leaves is high when you compare to the surrounding atmosphere. So then what happens? Diffusion occurs over there. Oxygen which is high in the leaves will get diffused out to the environment. Therefore, exchange of gases, that is carbon dioxide and oxygen, with external environment is diffusion. Osmosis. Now I have put a very simplified definition to understand what is osmosis. Do not go and see some complicated definitions and explanations. Please stick to this. Movement of water from the region of high water concentration to a region of low water concentration. Remember, here I am mentioning water concentration, high water concentration to a region of low water concentration through a selectively permeable membrane is called as osmosis. Suppose you keep a dry grape raisin in water. What happens? After some time you see that water is entered into the grapes and the grape has swollen up. That means the water has entered into the grape through the covering of the grape, which is selectively permeable membrane. It has allowed the water to enter into the grape. So, osmosis has occurred. Now, observe this diagram. Here, this red dot that you see is a solute molecule. Solute can be anything that is dissolved in the water, right? So, here it may be a salt or it may be sugar. And the blue part, the whole of this blue thing is the water. And in B section, you see the solutes are more when you compare to that of A. So what do you think? How will the water movement occur? And here you see this dotted line. This dotted line here, it is the semi-permeable membrane. So how do you think the molecules of water will move? The molecules of water will move from a region of higher concentration. So here in this A region, you have high concentration of water. So they will move from A to B so that they get to an equilibrium where both A and B have equal amount of water content. Suppose you place cell in hypotonic solution. What is hypotonic solution? That is, if you see our previous slide, the content where water is less, right, is hypertonic, and the side, that is A side, where water was more, is hypotonic. Here, if the cell is kept in a hypotonic solution, that is towards the A place, then what happens? Water will enter inside because water is more over here and here it is less. Since water is less in this cell and it is more in the outer environment, water will start moving from a region of high water concentration to a region of low water concentration, right? And when this happens, what happens? The cell will start swelling up. The cell will swell up. Now let's see here. The cell gains water if the medium surrounding the cell has a higher water concentration, that is hypotonic, than the cell. Now, if the cell is placed in isotonic, iso means similar. So if it is kept in isotonic solution, that is the water content in the surrounding as well as within the cell is same. Then what happens? The amount of water which is entering inside and the amount of water which is exiting out is equal. So there is no net gain or loss. So the cell will remain as it was. So let's see. The cell maintains the same water concentration if the medium surrounding the cell has same concentration as the cell. Water crosses the cell membrane in both the directions, but since the amount going in is same as the amount going out, the cell will stay the same size. The cell will stay of the same size. Now, if the cell is placed in hypertonic solution, what do we mean by hypertonic solution? The medium surrounding the cell, is having less water concentration as compared to the cell. Okay, that is the medium surrounding the cell has less water concentration as compared to the cell. So I told you, during osmosis, there is a tendency to attain equilibrium. That is, the cell will see that outside environment and inside environment is having equal amount of water. So the cell is always trying to see that this equilibrium is maintained. So the cell loses water if the medium has lower water concentration. Now I have put all these three diagrams together. When water content is more in the medium, then water will enter into the cell. When the water is equal in the medium, when you compare to the cell, then there is equal movement of water. The water that comes inside is equal to the water that goes outside. So the cell remains as such. If the water content is less in the outer medium, then the cell which has more water content will try to lose water, so that I told you it always try to come to an equilibrium with water. So it will try to lose water and it starts shrinking. Here are few more examples of osmosis. Unicellular freshwater organisms and plant cells gain water through osmosis. Plants' roots absorb water by osmosis. Molecular movement in and out of cell occurs due to osmosis. Let's now understand one more concept related to plasma membrane, that is endocytosis. Endo means within. Endocytosis is the process of transporting molecules into the cells by engulfing it with its membrane. The flexibility of the cell membrane enables the cell to engulf food, which we can see in case of amoeba. So here you see the diagram of amoeba, right? Here you see the cell membrane, since it is flexible. So the cell membrane is flexible, so it moves around and captures the food material which is here. So you remember the pseudopodia, it extends the pseudopodia and it takes the food particle inside and this gets covered. So this food particle moves into the cell. This process of taking inside the molecules or particles inside the cell is called as endocytosis. So if it is giving out, then it will be the opposite, which is exocytosis, right? It will be the opposite, which is exocytosis. Now let's come to cell wall. Cell wall is present in plant cells and fungal cells. It lies outside the membrane, which we have seen with help of diagram before. It is composed of cellulose. Cellulose you can see only in case of plants. This cellulose provides strength to plant cells. When a living cell loses water, there is shrinkage of contents of the cell away from the cell wall. This is called as plasmolysis. Now if you see this diagram that we have already discussed, you can see a cell, right? And when the living cell, this is the living cell, if it loses water, what happens? It starts shrinking away from the cell wall. This process of shrinking away from the cell wall is called as plasmolysis. The cell wall permits the cells of the plants and fungi and bacteria to withstand very dilute or hypotonic external media without bursting. So we saw this previously also. If you take a plant cell, the cell will swell up when it is kept in hypotonic solution. That is, water content is more on the outside. In this case, water will start entering into the cell and the cell will start swelling up. The cell wall will exert equal pressure against the swollen cell and it will not allow the cell to burst, right? But in case of animal cell, there is no cell wall and the cell will lyse, that is, it will break in hypotonic solution. So the cell wall prevents the osmotic bursting. In the next part, we will study about nucleus, cytoplasm, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles.